is 99% greater than 97%? Not necessarily if you're trying to compare sensitivities or specificities between two different labs' genetic tests. That's because there's no standardization on how to measure test characteristics in genetics. So when these numbers are provided by labs, their true meaning is often unclear. Now stick with me through this video because I've got some shockingly honest confessions from labs at the end. Let's take a step back and talk about why sensitivity and specificity even matter for patient care. Let's imagine you're in clinic and you suspect possible Lynch syndrome for a patient. Next, you order a comprehensive hereditary cancer test. You collect a saliva sample from your patient and send it off to the lab. The lab will isolate the patient's DNA and sequence the DNA. The raw sequence data is then processed by some intense computing referred to as bioinformatics, which produces variant calls. Some additional steps might also occur to confirm the accurate detection of variant calls. While your patient's variants are yet to be clinically classified, at this point, the analytical part of the test is complete. You want to make sure that the laboratory you're using can detect as many variants as possible in your patient's DNA. Analytical sensitivity informs us about the lab's ability to find variants. You also want to make sure that the laboratory isn't making mistakes by identifying variants that aren't actually there. Analytical specificity informs us about this possibility. The closer these numbers are to 100%, the better. Pro tip for remembering what these words mean. Sensitivity is 100% minus the false negative rate. Notice the letter N in both of these words. Sensitivity relates to false negatives, whereas specificity is 100% minus the false positive rate. Notice the letter P in both of these words. Specificity relates to false positives. Remember this, and it will help you keep these definitions straight. Now, it's true that additional metrics are needed to assess the true usefulness and accuracy of genetic tests. But as of April 2021, it's just analytical sensitivity and specificity that are the industry standards for describing next-generation sequencing tests. And as it turns out, these values are rarely cross-comparable between seemingly equivalent genetic tests from different labs. Here's how I know this. In 2018, the Association of Molecular Pathology, and CAP, published a systematic review of validations for tests which use next-gen sequencing. They found a clear absence of uniformity in the methods adopted for bioinformatics pipeline validation. They described that validation samples, number of samples, and data thresholds were highly variable, and that reporting of performance characteristics, i.e. analytic sensitivity, analytic specificity, and positive predictive value, was highly variable. Performance characteristics were not stratified by variant type in some of the studies. The reporting of confidence intervals for each of the performance characteristic measures was inconsistent among the reviewed studies. There's so much to unpack here, and so much that needs standardization across the industry. But let's focus on the finding that performance characteristics were not stratified by variant type in some of the studies. What does this idea of variant type mean for hereditary cancer tests? As it turns out, some pathogenic variant types are easy to detect with next-gen sequencing, and other pathogenic variant types are hard to detect. A study which will be published in 2021 by an international group of laboratories, led by a clinical lab here in the USA, has demonstrated that at least 10.3% of patients with disease-causing hereditary cancer variants have a variant that is hard to detect with short-read next-generation sequencing. This is the primary sequence technology used by most every lab doing hereditary cancer panels. To detect the hard variants, a lab has to invest a lot of resources into optimizing their sequencing chemistry and particularly their bioinformatics. But how many labs make this investment for their clinical genetic tests? The authors of this study took 13 hard-to-detect variants and ran them through 10 different laboratory workflows. These were labs who voluntarily signed up to be pressure tested in this very public way. Only 2 of the 13 hard-to-detect variants were detected by every lab. and only 3 of the 10 laboratory workflows could detect all of the 13 hard-to-detect variants. This matters for your patients because remember, at least 1 in 10 of the positive variants in hereditary cancer genes are hard to detect. Therefore, knowing which labs can detect hard variants and which cannot is super important. However, very few labs break down their description of analytical sensitivity by variant type. In fact, the authors of the study point out that some clinical labs intentionally stack the deck in their validation studies with easy variants so that their analytical sensitivities appear high, despite missing many pathogenic variants. But wait, you say, won't these labs be caught by the proficiency testing required by CLIA and CAP? The short answer is no. Labs can effectively choose the difficulty level of samples they use for proficiency testing, and most labs choose the easiest samples. The US government's CLIA Improvement Advisory Committee sent out anonymous surveys to next-gen sequencing laboratories. One of the many confessions was, quote, "Every laboratory and laboratory director is scared of failing a proficiency test," which means that laboratories want to pick the easy cases for proficiency testing. And quote, "There's no economic incentive for trying to look at more challenging cases. In fact, there is an economic disincentive, as well as the fear of failing a proficiency test." So no, proficiency testing, as it is typically utilized in the CLIA and CAP model, will not reveal which labs have used primarily easy variants to measure their analytical sensitivity. And while we have not gone into it here, there are easy mathematical ways to inflate analytical specificity. And it is not always transparent what math a lab has used to calculate their metrics. This whole issue of detection variability between labs is the reason why, when doing cascade testing in relatives for a known familial variant, it is considered best practice to order from the same original laboratory. For example, if my patient's brother was positive for a Lynch syndrome pathogenic variant, then I'll want to order site-specific testing from the lab that detected the familial pathogenic variant in the first place. And if I can't order from that lab, then I'll want to arrange for my patient's brother's sample to be sent as a control to the lab that my patient will be using. Most labs will run this positive control at no cost. If I don't do this, I might end up with a false negative for my patient and not know it. Since variant detection matters so much for accurate test results, we wish to encourage greater transparency from labs so that we know what variant types were used to validate their tests. Basically, how were the analytical sensitivities and specificities actually measured? With the support of the Utah Cancer Genomics Program of the Utah Department of Health and funded by the CDC, we are asking labs for more detailed descriptions of these metrics. We'll make these findings available at testwisely.org.